This is incubator. Cells are kept here to grow. It requires to be set at 37 degrees Celsius and 5% CO2. This device is called the centrifuge. Let's turn it on. Here we can set different parameters. We select revolution per minute and we adjust the required process time and then press start. Let's have a look inside the centrifuge. There are two different types of tube holders shown in green and brown for 15 ml and 50 ml tube. We are going to use 15 ml tube in order to do our experiments. Our materials are stored in two fridges, one at 5 degrees Celsius and the other at minus 20 degrees Celsius. This is called laminar hood. We perform cell culture experiment in here. This is a water bath. Let's turn it on. We have to adjust the temperature to 37 in order to cool down our material. We will place them here and leave them for some time. We need microscope in order to check the viability of our cells. In this case, microscope requires appropriate objective lenses to focus on live cells. To start our cell culture, we need to have our PBS, trypsin, and CCM, just complete culture media. We will place them into the water bath in order to cool them down. Let's take out our cells and check them under microscope. The subculture process is performed when the flask reaches to the confluence cells density 80 to 90%, where the entire surface of the flask is almost covered by the cell. This procedure needs to conduct in a laminar hood under sterile condition. Turn on the light, open the door until noise stops, wipe and spray inside the hood by 70% ethanol. After all, we can place all material inside the biological safety laminar, and to make sure you spray 70% ethanol before place them. We need a serological pipette, petri dish 10 centimeters and 15 ml tube. We'll make sure that we spray 70% ethanol before we place them. We move all of our materials, PBS, CCM, and trypsin, into laminar hood, and also we place our cells inside the laminar hood. We spray the vacuum tube. We need to culture our cells into other 3 petri dish. There are 10 centimeters and 50 ml tube. First, we need to remove the media. We are going to use the vacuum. I need to use the sterile tip. We'll take the medium out from one side, so we are not going to damage our cells. Anytime we are using the vacuum, we have to use the new tip. So this is the second time I'm going to wash cells with PBS. Because the medium will stop trypsin activity, so we have to wash cells and remove all the medium. For the removal of surface cells from the petri dish, 1 ml trypsin is added to the petri dish and it is incubated for 5 minutes to speed up the removal process. یکم اینجا رو مثلا ترش کن. So we add almost 15 ml CCM into new petri dish using surgical pipette. And also I'm going to prepare tube for centrifuge. We add 1 ml CCM into 15 ml tube. It is very important to label our petri dish, like write our name, date, and passage number. We don't need trypsin and PBS anymore, and we'll remove it back to the fridge. Cells are observed under microscope to ensure that all cells are detached from the surface of the cells and there is no cells aggregation. As you can see here, cells are detaching from the surface and there is no aggregation. Next, I'm going to add 1 ml CCM into petri dish in order to stop trypsin's activity. And now the mixture of cells, trypsin and CCM are added into 50 ml tube. Now we need to put our mixture, which already added into a 50 ml tube, to the centrifuge. Is centrifuge at 1500 RPM for 2 minutes. I'm using other 50 ml tube, which has the same amount of liquid as our mixture in order to keep the balance. So we need to take the supernatant out. Supernatant is the mixture of the trypsin and CCM. So we need to remove it. And resuspend cells with new CCM. So now we need to add the suspensions, mixture of the cells and CCM, into new petri dish that we prepared before. Now we are going to check the cells under microscope. There are flow cells there. Take out all discard materials from safety laminar and wipe inside of biological safety laminar hood by 70% ethanol. Close the door properly and turn on UV light.